Okay, so today I'm going to talk about the function and localization of human consciousness. Now, consciousness is a major unsolved problem in science. And even until today, scientists still have no idea how a physical system like the brain can ever have subjective experiences. And also due to this subjective nature of consciousness, this topic of consciousness research is once a forbidden topic, and especially in the time of behaviorism. But fortunately, in the 80s and 90s, a few things happened and that changed the situation of consciousness research. And the first thing is that Francis Crick and Gerald Edelman, so with these two Nobel laureates joining the study of consciousness and proposing reasonable ways to study consciousness, this research topic has now become much more visible and acceptable. And secondly, with new brain imaging technology like fMRI and new psychophysical tools, which I'll talk about later, and with these new techniques, the study of consciousness has now become much more objective and reliable. And the scientific studies of consciousness can be roughly categorized into two directions according to the question we ask. And the first direction is about how or where consciousness emerges in the brain. And in other words, it's about the mechanism of consciousness. And the second big direction of consciousness research is about why there is consciousness. In other words, it's about the functions of consciousness. And in today's talk, I'm going to tell you what we have done in my lab with regard to these two different questions. Now, first, let's look at the mechanism, or the question about how or where consciousness emerges in the brain. And to study these questions, most people use a paradigm called the Neurocorrelates of consciousness paradigm, the NCC paradigm. And this is proposed by Francis Crick and Christoph Koch. And the idea is quite straightforward. So they want to identify the minimum part of the brain that's sufficient, or that's enough for generating consciousness. So to study neurocorrelates of consciousness, there are basically two different ways. One is to study levels of consciousness. So like for example, right now we're totally awake, but when you go to sleep without dreaming, then you lose your consciousness. Or for example, when you're in vegetative states, then you lose consciousness. So when you change the level of consciousness like that, we can ask what happens in the brain. And the research method is quite straightforward. We can put you in the fMRI scanner and to see what happens in your brain when the levels of consciousness change from totally awake to totally unconscious. Okay, and a different way to study neurocorrelates of consciousness is to use things like bistable illusion. And in other words, it's like, well, right now you're totally awake, but if I change the stimulus, usually the contents of your consciousness will change. So we can ask, when there is a fluctuation of conscious contents, what happens in the brain? And now I'm going to tell you, I'll give you some example in my lab to tell you how we study consciousness scientifically in the lab. So now let's look at the first way of studying neurocorrelates of consciousness, which is to study the levels of consciousness. So in the past few 20 or 30 years, it's indicated that, well, one of the critical structure in the brain is this thing called the thalamocortical connection, which is the connection between your thalamus and cerebral cortex. And it seems that this structure is quite crucial for whether you can have intact consciousness or not. And let's take this one, for example. So this is a study by Stephen Laurie. Excuse me, I can see the figure. Okay, by Stephen Laurie, and they discover so for example, let's take the green dots here, for example. So the green dots here shows the control subjects. So each one dot is one control subject, meaning normal healthy subjects. And you can see that the correlation between thalamus and prefrontal cortex is there's a positive correlation. However, for those people who's suffering from vegetative states, you can see that the thalamus and prefrontal cortex, they have negative correlation. But some of these people, some of these vegetative state people, when they recover from this state and become normal again. So you can see that after recovery, the blue stars or blue dots shows that the correlation between thalamus and prefrontal cortex become positively correlated again. So it seems that there's a, when you have intact consciousness, usually the correlation between thalamus and prefrontal cortex is positively correlated. But when you lose consciousness, it becomes negatively correlated. And here's another review from about sleep. You can see that across many studies, when you enter the sleep stage without dreaming, so presumably without consciousness, you'll see thalamus activation, but cortical deactivation. So once again, showing that there's a negative correlation between thalamus and cortex when you lose consciousness. And here's another example about anesthesia. So you can see that across many different studies using different kinds of drugs, when you lose consciousness under drugs, you'll see that there's a negative correlation between thalamus and cortex again. So once again, you'll see that there's a positive activation in the thalamus, but negative activation in the cortex. So once again, showing that this connection between thalamus and cortex is important. Okay, so in my lab, we're interested in this thalamocortical connection and its relationship to consciousness. So we decided to look for other possible phenomenon or conscious phenomenon that so that we can study this thalamocortical connection. And the phenomenon we decided to study is something called microsleep. So microsleep is referring to brief episodes of sleep-like states, and that are often accompanied by behavioral signals such as eye closure or attentional lapses. And in fact, everyone should have this experiences, and actually you should be quite familiar with it, because this is the phenomenon that usually happens when you are attending a boring talk, especially in the afternoon. So when you cannot attend anymore, usually you see this microsleep happening. Okay, and it's worth noting that microsleep does not equal to early sleep stage, right? Because sometimes people will think that, well, microsleep is probably just early sleep stage, just the beginning of the sleep stage. But we don't think this is the case. And this is because in the literature, we discovered that in the early sleep stage, usually you see brain activation to decrease. And this can be found with brain imaging or fMRI. So whenever you see that, well, a subject fall into a sleep, you'll see that the brain activation will decrease. In contrast, in microsleep, cortical activity usually will increase. And you can see that, well, in a few recent studies, they discovered that when you enter this microsleep, you will see cortical activation instead of cortical deactivation. So it seems that it's the opposite of early sleep stage. Okay, so we discovered that while in the literature, there's this widespread activation during microsleep. So we're interested in knowing why this is the case. So we actually come up with two hypotheses. The first hypothesis is that, well, during microsleep, these brain activation across the brain, it could be, for example, due to auditory stimulation. And for example, maybe when you enter this microsleep, you are still processing auditory information, and maybe this information is processed and sent across the whole brain, and that's why you see the whole brain activation like that. And another possibility is that, well, maybe these activations have nothing to do with auditory information processing. Maybe that's more like a default state when you fall into microsleep. You tend to have like this widespread brain activation, whether you have auditory information processing or auditory signals at all. So we decided to test this two hypotheses. And one way to test that is simply that we can present different kinds of auditory information when people fall into microsleep. And our idea is very simple. If the people are still processing auditory information in microsleep, then we should be able to differentiate their brain activation with respect to these four different sounds. However, if people in microsleep don't care about what kind of auditory information are there in the environment, then no matter what kind of information we play when they're in microsleep, their brain activation shouldn't be affected. Okay, so here's the result. So we discovered that during microsleep, there's wide range of sensory and motor areas activated. And this is what happens in the brain during microsleep when we did not play any auditory information. So you can see that even without any auditory information, there's a widespread cortical activation already. And second, when we played the auditory information during microsleep, you can see that the activation is quite similar. And that's another indication showing that the auditory information doesn't affect this activation too much. And when you take the contrast of stimulus on minus stimulus off, you can see that the activation is limited in auditory cortex only. So these data are basically showing that while during microsleep, these activation has probably nothing to do with auditory information. And if auditory information is playing any roles, is limited in the auditory cortex. And here's another way to analyze the data. So we can, because we have 4 kinds of different sounds, so we can take the brain activation responding to the 4 different kinds of sounds and run a similarity check. And you can see that when the subjects are awake and when you play the sounds, you can see a similar activation when the pitch is the same. So for example, when the pitch are both high, the brain responds similarly. And when the pitch are both low, the brain responds similarly. And what is most interesting is that, well, during microsleep, when we play the sounds, no matter what kind of sounds we play, the brain responds very similar to each other. Okay, and also most interestingly, when we do not play any sounds during microsleep, the brain activation is still similar to those brain states when the sounds are played. So in other words, no matter what kind of sounds we play, or no matter we play any sounds or not, the brain during microsleep responds very similar to each other. Okay, so a quick summary about this first study. We discovered that microsleep involves little auditory information processing. So if there's any auditory information processing, it's also limited to auditory cortex only. And second, we discover stimulus invariant brain activity patterns during microsleep. And for now, we are not sure why these brain activation patterns are stimulus invariant. And we are suspecting that this is probably due to efforts of trying to stay awake. Because we know that while during microsleep, people are fighting against their desire to fall asleep. So maybe it's this effort of trying to stay awake that's causing this widespread brain activation during microsleep. And most importantly, our data supports that consciousness is related to thalamocortical disconnection, because we discovered that during microsleep, presumably when you lose consciousness, there is a activation in the cortex, but there is a deactivation in the thalamus. So this is consistent with previous finding showing that consciousness is related to thalamocortical disconnection. Okay, so that's an example of how we study levels of consciousness and its neurocorrelates in the lab. And in the rest of the talk, I'm gonna do something similar. I'm gonna give you some example in each direction, showing that, and to tell you how we study consciousness in the lab. And if you have any questions with regard to any particular study, feel free to interrupt me anytime if you want. And I'll be happy to just answer your questions before the Q&A session. Okay, so now let's move on to the second direction, which is another way to study neurocorrelates of consciousness. Okay, so this way of studying neurocorrelates of consciousness, usually people will use a paradigm called bistable illusion. And if you've never seen a bistable illusion, here's one example called Necker cube. So in Necker cube, you see a cube like this. And usually you will have at least 2 different kinds of percept when you're observing this, this figure. One is something like this. You'll see that this cube is almost like sitting on a plane that's horizontal here. And another possibility is that this cube is probably sitting or attaching to the wall, like here. So it's facing to the other direction. And if you simply look at this figure, and you'll see that your percept will start to switch between these two different possibilities. And a good thing of this illusion is that the stimulus itself is always identical. So the stimulus itself never changed, but the percept in your mind or in your brain is constantly flipping between two possibilities. So we can take advantage of this illusion and simply put people in the fMRI scanner and ask them to report their perceived percept. And in the meantime, we can observe which part of the brain correlates with their percept. So therefore, we can identify the so-called neurocorrelates of conscious perception in this way. And here's another example. You see that there are two faces, or there's one face or one vase here. And once again, this is a bistable illusion. So we can put people in a scanner and see which part of the brain changes together with their percept. And here's another example called perceptual fade. And once again, you can see that there's a red dots here. If you fixate, you stare at the red dots and don't move your eyes, you see that the gray ring will very soon, it will disappear from your consciousness after a few seconds. And once again, this is a bistable illusion because, well, once you move your eyes, the percept will come back. I'll give you one more example. This is something called motion-induced loneliness. So you can see that there's a yellow target here, but if you fixate, if you stare at the white dots, now, if you don't move your eyes, and when I turn on the motion backgrounds, you'll see that the yellow dots will disappear from your consciousness very quickly after a few seconds. But this is an illusion because the yellow dots actually is always on the screen. It never physically disappeared. So once again, this is a bistable illusion because, well, sometimes when you move your eyes, the target will come back into your consciousness again. So it'll just go disappear and reappear like on and off. Okay, so and in the past few years, my lab have discovered that neurocorrelates of visual consciousness, in this case, can be found in V1 in most of the cases. And for example, when we use motion-induced blindness, we discover that when a target disappeared, you'll see that the activation in V1 will decrease. But when it reappears into your consciousness, the V1 activity will increase. And you see that there's a nice correlation between your consciousness and V1 activity in this case. And also when we use perceptual fading or perceptual filling in, once again, we discover that when the gray ring disappears from your consciousness, you'll see V1 activity decrease. When the gray ring reappear into your consciousness, you'll see V1 activity increase. And I'll give you another example. In this example, we use something called the Muni Images. So in this case, we show people first about these images called Muni Images. And this is created by a person called Muni. That's why it's called Muni Images. And you can see that while most people, when they see this picture, they cannot understand what it is in the very beginning. And here's another example. When they first see this picture, they don't understand what it is. But when we give the participants or these people, when we tell them what this picture is by showing them the grayscale photo of that same picture, and now people can see that this is a frog and this is a fish. So after this kind of one-time priming, people will start to see the original, see the object in the original picture. So once again, so here, if you show them the original Muni picture again, they will start to see that this is a frog and this is a fish. So our question here is very straightforward. We want to know that, well, before you understand what this picture is, we show this picture to you and your conscious contents would be just black and white blobs because you don't understand what it is. And now if I tell you, and same is true for your higher level neural representation and lower level neural representation. Now the question is, well, if I tell you what it is, right? If I show you that this is a camel, and right away your conscious content will change. You start to see this picture as a camel. And now we want to know what happens in the brain. So for example, does your higher level visual neural representation change after you know what it is? And how about your lower level visual neural representation? Will it change and after you recognize this picture? So to answer the questions, we pre-identified a few regions of interest, including LOC. This is a higher level object recognition area called lateral occipital complex. And we also identify another area called foveal confluence. This is the foveal part of your visual cortex, V1, V2, V3 combined. So this is one represent the higher level visual area and one represent the lower visual area. And the procedure goes like this. So we will bring in subjects and we make sure that they couldn't understand what this picture is. And we show them these pictures and they have to do an attention task by answering whether this picture has high or low contrast. Like this is high and this is low contrast, high contrast. So they have two buttons to report the contrast of the picture. Now, after 30 minutes, we collect the brain data and then we start to show the grayscale photo of this picture. In this case, you can see that this is camel. And once again, they're doing an orthogonal task by answering the contrast of this picture, high, like low contrast or high contrast. And after another 30 minutes, we started to show them the original Muni picture again. And once again, they're doing the orthogonal contrast task. Okay, so after 1.5 hours, we collected 3 stages of their brain data. So we have the before stage, we have the photo stage, and we have the after stage. And our question here is very simple. We want to know, after they know the interpretation, after they recognize this picture as a camel, does the neural representation change in their brain? Okay, and we use an analysis called multivariate pattern analysis, MVPA, and it's quite straightforward. For example, we can take an area, and we know that this area is composed of many different voxels. So we can simply check the activation pattern of this area. So by examining the activation pattern in this region, and then we can ask, well, what happens in the brain in this area after you know what it is? And our hypothesis is that, well, we think that after the people know the interpretation of this picture, the neural representation should change and become more like the representation when you're looking at the grayscale photo. So if this hypothesis is correct, we expect to see that the correlation or the similarity between after and photo should be greater than the similarity or correlation between before and photo. Okay, now let's check to see if the results fits our hypothesis. So we can see that in this higher level area, LOC, you can see that the red bar represents the correlation between before and photo, and the green bar represents the correlation or similarity between after and photo. And you can see that the similarity between after and photo is significantly greater than the correlation between before and photo. Suggesting that after you know the interpretation of the picture, the activation in LOC, the activation pattern will change and become as if you're looking at a grayscale photo. And now how about early visual area? So you can see that the red bar here represents the correlation between before and photo, and the green bar here represent the correlation between after and photo. And once again, you can see that the correlation between after and photo is significantly greater than correlation between before and photo. So once again, showing that while after you know the interpretation of this picture, the neural activation pattern in early visual area will change and become more like as if you're looking at the grayscale photo of that picture. And in experiment 2, we tested a bunch of different kinds of Muni pictures. And we also changed the paradigm a little bit. So we reduced the presentation duration from 500 milliseconds to 180, trying to minimize eye movements compound. And we also changed from block design to event related to try to control for anticipatory signals. And once again, the results replicates our finding, showing that while after you know the interpretation of the picture, the activation of the pattern in early visual area will change and become as if you're looking at a grayscale photo. Okay, so in this second study, we basically discovered that after you know the interpretation of the object, the content of your consciousness will change. And so is true for your higher level neural representation, and so is true for your lower level neural representation. So once again, this is an example showing that neurocorrelates of visual consciousness can be found in V1. And we are not the only lab or the first lab to discover this. And actually, there are many different labs using same paradigms or different kinds of visual illusion or different kinds of paradigm. And they all discover that in most of the cases, neurocorrelates of consciousness can be found in V1. And this is suggesting, or this is supporting a debate about whether consciousness or visual consciousness is realized or correlating with the frontal part or the later part. And these findings is actually supporting the view that neurocorrelates of consciousness is more realized in the posterior part of the brain. And I'm happy to talk about this debate more later in the Q&A session. Okay, so now, so you just saw a few other examples of how we study neurocorrelates of consciousness, especially about the fluctuation of visual consciousness in the brain. And now I'm going to move on to the other direction of consciousness research, which is about the functions of consciousness. Okay, now with regard to the functions of consciousness, there are actually many different kinds of theories in the field, in the history. Like, for example, some people claim that the function of consciousness is for providing more unified representation. And some people are claiming that the functions of consciousness is about providing more global information, and maybe it's related to more flexible control, and it's also related to self-awareness or social coordination. However, there are so many different claims or hypotheses or theories about functions of consciousness, but it seems that there are not many solid way to test this hypothesis. So in the lab, we think that, well, it should be one of the most important thing is actually to design experiments to test these different theories or hypotheses. So to do so, we are proposing a way to test the functions of consciousness by doing something called consciousness knockout or knockdown. And the knockout, or this term is borrowed from molecular biology, because, well, usually they would knock out a gene or knock down a gene to see whether this gene is responsible for certain functions or not. And we're proposing that we should do the same thing. We should try consciousness knockout experiment to see when you knock out consciousness, whether these functions still survive or not. And if the functions of consciousness is really about these things, then when you knock out consciousness, you should see that the functions of these claimed activities should drop. And if consciousness is turned out to be has nothing to do with these functions, then when you knock out consciousness, these functions should still survive. So that's how we're going to test whether consciousness has these functions or not. So I'm going to give you one example called visual pop-out. So visual pop-out is a phenomenon that when you have a feature singleton, like red among a bunch of green, then this feature singleton will just pop out immediately and automatically from your visual awareness. You're going to detect this red thing very quickly and automatically. So this is something called visual pop-out. And we know that this phenomenon is very robust. It's very fast and very automatic, but we want to know how automatic and how fast can it be? And in other words, we are asking, well, do we need consciousness to have this kind of information processing or not? So in other words, we are asking, can we process invisible saliency or not? In other words, well, if you have no visual consciousness about this stimulus, is it still possible that this feature singleton can still be processed unconsciously and attract your attention? So we want to know these questions. And of course, then to answer the questions, we need to knock out your consciousness and see whether this feature singleton can still attract your attention or not. And now you must be wondering, so how are we going to knock out your consciousness about this stimulus? And here's how we do this. So we're using the paradigm called consciousness knockout paradigm, or something called continuous flat suppression. And this is a paradigm developed by Untitled Chapter and Christoph Koch in the around 2005. And the method is something like this. You can present something that's constantly flashing into one eye through this mirror stereoscope. And in the other eye, you can present something static that you don't want your you don't want your participant to see. So in this case, because our brain is much more sensitive and to these constantly moving things. So if you ask your subject what they perceive in this condition, they'll tell you that this changing colorful squares are the only thing that I can perceive. And if you ask them, well, do you perceive any colorful or any red or green dots in the other eyes? They'll tell you that they couldn't see it. And this kind of continuous flash suppression can last for sometimes many seconds and up to minutes. So this is basically the way we knock out people's consciousness about this display. And given that, we can still ask, well, given that the subjects did not perceive any information in this eye, but does this feature singleton still attract their attention or not? So our hypothesis is that, well, even when subjects did not perceive this stimulus, the feature singleton can still probably be processed and enhance visual sensitivity at that location. And this is how we test the idea. So we bring in subjects and we show them flashing, changing colorful squares in one eye. And in the other eye, we present a static pictures containing a feature singleton. And of course, the subjects would say that, well, this is the only thing that they perceive, and they did not see anything about this, these green or red stimulus. And after a short blank, we started to measure their visual acuity at the location that has been primed or at the location that has not been primed. And the visual acuity is measured by an orientation discrimination task. So you can see that this Gabor patch is slightly tilted to the left or right by 1 visual degree. And subjects have to respond whether this Gabor patch is tilted to the left or right. So it's a left-right orientation discrimination task. And to make sure that the subjects did not see this display, so we have them report whether they see anything about the green and red dots or not. And if in any trial they reported they actually see something, we would abort a trial just to use the data that they are totally unconscious about a stimulus. And there are a few factors that are randomized and counterbalanced. For example, the suppressed eye is sometimes left, sometimes right. And the feature singleton location is not always here. It could be in one of the 12 locations on this ring position. And the tilt direction of the Gabor could be left or right. So everything is counterbalanced and randomized. And here's the result. So we discovered that, well, even when subjects are claiming that they did not see anything about this display, but when you measure their visual acuity is actually higher and more accurate or more correct at the location that has been primed by this feature singleton, comparing to a control location that has not been primed. So you can see that, well, even when subjects did not see anything about this stimulus, but their visual acuity is still being affected by this unconscious target. So in other words, unconscious saliency can still be processed in this scenario. Okay, and after we discovered this finding, one of my students, Sean, he decided to test other kinds of information to see if we can process these information unconsciously. So he tested a few things like syntax, facial attractiveness, and sound shape mapping. So for example, and basically he's using the same paradigm. He's using continuous flash suppression. And a little bit different is that he is actually measuring the time it takes to break. So for example, in this experiment, he, in the unconscious eye, he put in 2 different kinds of possible sentences. One is a syntactically congruent sentence, like birds eat worms. So this is a typical syntactical sentence composed of, there's a subject and there's a verb and there's a noun here. And but this sentence can also be syntactically incongruent, like birds eat drink. So you have 2 verbs here. So it's syntactically incongruent. So in this unconscious eye, there are these 2 different possible sentences. And what he measured is the time it takes for people to see these sentences. And as I said before, during continuous flash suppression, although the suppression can last for many seconds and sometimes up to minutes, but eventually it will start to break. And we are actually measuring the time it takes to break. And you can see that in this case, the syntactically incongruent sentence can actually break consciousness much more faster or significantly faster than syntactically congruent sentence. So in other words, even though in the beginning, your brain is not consciously perceiving these sentences, but your brain is still unconsciously processing these sentences, so that one of the sentence or one of the condition will be processed much more efficiently and enter your consciousness much faster than the other condition. And here the same, in another experiments, he put in two different kinds of faces. One is more attractive and the other one is more unattractive. And he discovered that while the attractive face will be processed much more faster than the unattractive face. So in other words, the message is the same. Even when in the beginning, you cannot see any of the stimulus, but your brain is still unconsciously processing this stimulus, so that one kind of the stimulus will be processed more efficiently and will be finished earlier and enter your consciousness earlier. And here's another example. So in this experiment, the third experiment here, he tested sound shape mapping. So this is a phenomenon that has been found in psycholinguistics. So for example, if I ask you to name these two different shapes, which one would you call it booba and which one would you call it kiki? And it has been discovered that in most cases, people would tend to pair this round shape with this round sound booba. And they will pair this spiky shape with this spiky sound kiki. And this is probably because, well, people tend to pair features that are similar together. But we're interested in knowing this kind of pairing, is it happening consciously or unconsciously? Because one possibility is that, well, we're doing this consciously, right? Because you're seeing that this is round and you're feeling that this is round as well. So therefore you think the two should be paired together. But another possibility is that maybe this pairing is happening unconsciously. So maybe your visual and auditory system is they're processing this information unconsciously. So the pairing is happening even when you are unconscious about the stimulus. So to test this hypothesis, we simply put in stimulus in the unconscious eye. And when there are two kinds of different stimulus, one is a congruent one. So you see that there's a round shape with a round sound. So this is congruent. And another possibility or another stimulus is spiky shape with round sound. And we discovered that, well, when you present these two different kinds of stimulus unconsciously, it's actually the congruent one that will be processed faster and enter your consciousness earlier. But once again, the information is the message is the same. So it seems that our brain is able to process information unconsciously, so that some kind of stimulus will be processed more efficiently and enter your consciousness earlier or faster. All right, now I still have a few times, so I'm going to talk about the current and future directions in my lab. And the first direction is that, well, if unconscious information can influence behavior, like I just show you, so there are many unconscious information processed in the brain, and it seems that these information, even though unconscious, can still influence your behaviors. So if this is true, is it possible that we can localize the corresponding brain areas? Is it possible that we simply go into the brain and identify these unconscious information? And furthermore, can we use these unconscious information to predict people's behavior by simply reading these signals and predict people's behavior? So to test this question, to examine these questions, the first experiment we did is something called binocular rivalry. So binocular rivalry occurs when your two eyes receive two different inputs. Like in this case, your left eye is receiving a house and your right eye is receiving a face. And in this kind of scenario, your brain will get confused and start to try to figure out what's going on. So usually your brain will try to resolve the ambiguity and give you one possibility at a time. So as a subject in this scenario, you'll see either face or house. And the percept will start to switch between face and house every few seconds. And here's a simple demo of this binocular rivalry by using this superimposed image. So you can see that there's a face and there's a house. If you just stare at this picture and attend into one of it, you can see that one of the stimulus will become more clear. Like for example, if you attend to the face, you'll see that the face become more clear and the house will become like suppressed. And the opposite is that, well, if you attend to the house, you see that the face image will become suppressed. So this is a simulation very similar to binocular rivalry. Okay, and in our experiment, what we're interested is that while each time when we present a stimulus like this to the participants, they either see it as a face or house, okay? And if you do this for 1,000 trials, you'll see that, well, half of the time, there are 500 trials that they perceive it as a face, and the other 500 trials, they perceive it as a house. But we're curious why this is happening, because stimulus is always the same. So there must be something in the brain that's determining whether you're gonna perceive a face or house. And our hypothesis is that, well, there's probably some signal in the brain that's biasing your perception. And if this is true, we should be able to use something like fMRI to capture this information and even use this information to predict whether you're going to perceive face or house in the next trial. So this is exactly what we're going to do. So we are going to present stimulus like this to the subjects. And before the stimulus onset, we're going to examine their brain activation to see if there's any pre-stimulus activity in the visual cortex that can predict the subsequent percept. And here is the procedure. So basically we let them wear red and green filters in the fMRI scanner, and we present stimulus like this to them. So every 2 seconds, there's a stimulus and they have to respond whether they see a face or house. And sometimes we will present a yellow face so that they're guaranteed to see a face like this one. And sometimes we'll present a yellow house. And once again, just to guarantee that they will perceive house in this trial. And we have two regions of interest. One is something called FFA, this fusiform face area, which has been implicated in face processing. And there's another area called parahippocampal place area, indicated in place or scene processing. And we also identify foveal confluence, representing the early visual area again. And all these areas are pre-identified. So we collect these areas before the experiments. And we are especially interested in FFA and PPA because the stimulus we use is containing a face and a house. So we think that, well, if there's anything that can bias their percept, it's probably going to be the face area and the scene place area. Okay, so here's how we analyze the data. So this is one example run. So you can see that there are ambiguous stimulus, and sometimes there are non-ambiguous stimulus or non-rivalry stimulus. And in this example run, people might see face in the first trial, house in the second trial, and face in the third trial, house in the fourth trial. And for these control stimulus, they must see face and house. And now let's look at the data for the non-rivalry stimulus first. And you can see that in the area FFA, this is the area that responds more to faces. You can see that when you present a face, their response is higher comparing to when you present a house. And in PPA is the opposite. And in the Foveal Confluence, the early visual area, it seems that it's responding more to the houses, and probably because there are more high spatial frequency information in the houses. And these findings are as expected. There's nothing surprising here. And it's replicating previous findings showing that FFA is responding more to faces and PPA responding more to houses. And it's showing that we have enough statistical power to pick up differences that are bound to be there. Now, of course, the key information or the key analysis is what's happening in the brain before you see these rivalry stimulus. And we want to know, does pre-stimulus activity in the brain predict the subsequent percept or not? And to answer these questions, we did a simple analysis. We simply take those trials that are perceived as face and separate from all other trials that are perceived as house. And we simply analyze whether there's a difference in pre-stimulus activity between these two kinds of condition. And you can see that this is in the FFA, and 0 is the time of the stimulus onset. And you can see that when an ambiguous or a rival stimulus is perceived as a face, FFA responds more. And when this stimulus is perceived as a house, it responds less. And this is, again, not surprising. This has been shown before, but what we're interested is what happened in the pre-stimulus period. And you can see that before the stimulus onset, basically the mean activation is flat, it's 0. But don't forget, this is a conventional way to analyze fMRI data. So basically you take a region and you take the mean of that area. But what if we don't take the mean and we keep the pattern information? And you can see that if this region actually responds differently to face and house in different pattern, now if you take the mean, the signal will disappear because the mean will be the same. So one way to keep the information is not taking the average, but use pattern analysis to run the analysis, to run the study. So in here, we basically use Huxley correlation. So we split the data into two halves, and we check whether this area responds differently to face or house. And if it's responding differently to face and house, we should expect to see that the within category correlations should be stronger than the between category correlation. So we can take the within minus between, and if the value is greater than 0, then we know that this area is responding differently to face and house. And another possibility is that maybe this area doesn't care about whether it's a face or house. And so in that case, if you do within minus between, the value will be 0. So basically, the greater this value, the more difference this area is responding to face and house. And here's the result. So we show that while 2 seconds before the stimulus onset, actually FFA is already responding differently. So there are 2 different patterns before the stimulus onset. And it seems that when one pattern shows up, then later the subject is more likely to perceive a face. And if you see another pattern 2 seconds before the stimulus onset, then later that trial will be more likely to be a house. And this is only found in FFA, but not in PPA and Foveal Confluence. And we show that this is not due to contamination from the previous trial, because when we analyze the data, we make sure that the trial history is balanced. And we also, and please note that there's no mean bold difference, but only pattern difference, and suggesting that there's probably endogenous variation in this area that are involved in determining which category will be perceived. Okay, so and what you just see is actually a lower level perceptual decision. So after this study, we started to think that, well, how about higher level decision-making paradigm? Is it possible that we can still predict people's decision in these other kinds of higher level decision-making paradigm? So to answer the questions, one of my previous graduate students, and he decided to test or to examine the neural antecedents of decisions by doing the following experiments. So he simply presented a picture like this to the subjects, and the subjects have to say whether they like it or dislike it. So it's a very simple decision-making paradigm. You see the photo and you answer whether you like or dislike. And we have hundreds of different photos like that. So after each one, you have to say you like it or dislike it. And before that, for each picture, we collected their brain activation pattern 16 seconds before the stimulus onset. And we discovered that actually 4 to 6 seconds before the stimulus onset, there's already pattern difference in certain brain areas, including medial frontal gyrus and anterior cingulate cortex. And you'll see that in these regions, actually there are 2 different kinds of pattern. And when you observe one pattern, then later people are more likely to say they will like the picture. But if another pattern shows up, then people are more likely to say that they will dislike that pattern. So it seems that we can use this signal to predict whether people are going to like or dislike a subsequent pictures or not. And here's another paradigm that we use at different kinds of decision-making paradigm. And in this case, it's a risk decision-making paradigm. So in this case, for example, we give them 2 options. One is that you have 100% chance to get $4. But if you choose this option, you only have 50% chance to get $8, but you have 50% chance to get nothing. So you can see that the expected value is the same, but if you're more conservative, you'll probably pick this one to get $4 for sure. But if you're a risk taker, then you probably want to gamble your luck to try to see if you can get $8 in this case. And again, we have hundreds of trials like this, and the expected value is more or less the same. And in each trial, the participant has to have to decide whether they are going to gamble or not. And once again, we collect their brain activation 16 seconds before the stimulus onset. And we discover that 4 to 6 seconds before, there are a few regions that already show a difference. There are basically 2 different kinds of pattern in these regions. And when one pattern show up, then later they're more likely to gamble. But if you see another pattern, then a few seconds later, they're more likely to not gamble. So once again, we can read out these brain activation patterns and predict whether they're gonna gamble or not in the subsequent trials. All right, so in this direction, we basically show that yes, there are unconscious information that can influence behavior, and we can actually use brain imaging tools to localize these information and use it to predict people's behaviors. Okay, and the second direction that we're currently working on is that, well, if there's unconscious information that can influence behavior, then is it possible that we actively change people's behavior unconsciously by inserting certain unconscious information into their brains? And I'll give you one example that we're currently working on. So this is an example, a paradigm that we're hoping to do. So we are hoping to read people's brain activation when they're sleeping. And if we detect certain kind of patterns, let's say if they're having nightmare, then we're hoping that we can interfere and change their brain activation or dream contents to alleviate this nightmare symptoms. And here's how we are going to do. So basically, we're going to scan people when they're sleeping. And we are going to use a serial awakening paradigm. So like, for example, an 8-hour sleep, we're going to wake them up every now and then to see whether they're sleeping or not. And therefore, collecting their brain data corresponding to brain, dreaming or no dreaming, and therefore building up this database. And you can see that this is the preliminary data. So this is the brain activation pattern using EEG when we discover they're dreaming. And when they're not dreaming, this is the brain activation pattern. And you can see that by eyeballing it, there's already a difference in the, especially the low spatial, the low frequency range. You can see that there are more activation in the low spatial, low frequency range when they're not sleeping. And basically we can simply, by reading their EEG data in real time, and we can already differentiate whether they are dreaming or not. And our next step is we're trying to see if we can further differentiate what kind of dream are they having, and especially whether they're having positive dream or negative emotional content dreams. And our idea is that we are gonna further differentiate their dream states into positive and negative emotional dreams. And if we see negative events or negative dream happening, we're going to make some interference, trying to, let's say, for example, wake them up or play some smooth music or sentences or language to try to alleviate their nightmare symptoms. Okay, and finally, the last approach that I'm going to introduce you is that, well, we're hoping to build a consciousness meter. For example, and this is quite useful because, well, if we can have a consciousness meter, we should be able to measure the degrees or levels of consciousness in patients, and for example, in fetus or in animals. And for the patients, we can probably use this methods to predict the recovery likelihood of them. And of course, we are not the first one to try to do this. And for example, this is a study by Adrian Owen in 2006. They discovered that, well, neuroimaging can be used as an objective tool to supplement behavioral tests of these vegetative state patients. And you can see that, so in this case, they bring in the patients and ask them to imagine playing tennis or imagine doing spatial navigation in their house. And you can see that in this vegetative state patients, after they're receiving these instructions, their brain responds as if their brain responds very similarly to control subjects. So indicating that they're probably still conscious in this case and can take your instruction and follow your instruction. Okay, and then we're interested in this finding. So we decided to dig further and investigate what are the paradigm they use exactly. And you can see that in this standard fMRI command following paradigm proposed by Adrian Owen, they usually use something like this. So like imagine playing tennis versus imagine walking around in your room. And they're suggesting that 10 minutes scan is enough because, well, they're saying that, well, if you scan them for too long, then people tend to get fatigued. And then you have signal get washed out by these non-responding periods. So 10 minutes is enough. And also they're saying that, well, with their paradigm, their standardized paradigm, they can discover up to 30% of responders, so which is very good. But we're not so convinced. So we started to question first, are these the best instruction or not? Because, well, it seems that it seems to us that this is probably not the best instruction because some people don't play tennis. And some patients, they've been away from their home for so long and they can't even remember the spatial layout of their rooms. So maybe there are other even more simple instructions that can be used. And second, is 10 minutes really long enough? So we're suggesting that, well, maybe if we can scan for a much longer time, perhaps we can get much better signal. And finally, we're asking, well, is this really a good prediction for recovery? Because we have, because it seems that, well, 30% of responders is way higher than those people who can actually recover. So maybe this is not a good prediction of recovery. And to answer these questions, we started to change the paradigm and simplify it. So for example, we changed it into a much simpler command following paradigm. And we decided to scan for much longer. And the paradigm we changed is something like this. We use kick left leg versus punch right fist. So we presumably this is much simpler than imagine playing tennis or walking around in your house because everyone knows how to kick leg or punch fist. And we decided to scan for much longer, sometimes up to 30 minutes or up to an hour to see if we can get better signals. And we have so far scanned 2 healthy controls and 13 vegetative state patients. And this is collected from 9 patients scanned for multiple times. And for example, I'll give you some data to look at here. So this is patient 1, we scanned for the first session. And patient 1 scanned for another session, and patient 2, session 1, patient 2, session 2, patient 2, session 3. And there are other patients. And each brain figure here is the average of 3 runs with 3 minutes each. So this is basically the mean of 10 minutes. And we can do a running window analysis to show you how the brain responds every 10 minutes across time. And you can see that this is basically the brain activation across time. And one interesting finding is that we actually discovered fluctuations of brain activation across time. For example, in this patients, in the beginning, there's no brain, they're not responding to the instruction. But in the third time window, they started to respond to the instruction in the motor areas. And for example, in this patient, session 2, in the beginning, they're responding, but later they stopped responding. And you can see this fluctuation basically can be found in every one of the subjects. Okay, and here's another way to show the same data. So the black shows that there's an activation in the corresponding motor areas. And once again, you can see that there's a fluctuation of on and off in motor area. Okay, and another interesting finding is that while actually some of the patients, although they are in vegetative states, but you can see that they still have this sleep-wake cycle. So in the daytime, they tend to open their eyes and appear to be more responding to the surrounding. But sometimes they'll close their eyes and appear to be not responding at all. And we actually documented their arousal level. So you can see that for those time that has an underline green line, it's actually meaning that they have very low arousal states. And interesting is that, well, sometimes when they're in this very low arousal states, you can still observe brain activation responding to the instruction. So we therefore have suspecting that maybe these kind of brain activation responding to the instruction is probably just unconscious information process. And I'm happy to tell you why we think so later in the Q&A. Okay, so basically in this study, we discovered that while are these the best instruction? The answer is no, because we discovered that we can use much simpler instruction and discover stronger signal. And once again, 10 minutes is not enough. We discovered that if you scan for much longer time, you can actually find better signals. And is this a good prediction for recovery? The answer is once again, no, because in every one of our subjects, we can discover that sometimes their brain will respond to the instruction. And since everyone is a responder, so it's unlikely that everyone will recover, right? So it cannot be a good prediction for recovery. So the summary of this last study, so basically we discovered that neuroimaging may provide an objective measure to supplement behavioral tests, but it can only be used to identify patients who can still process information, and probably they're probably they're processing it unconsciously. And this tool is not ideal for predicting recovery or predicting consciousness. Okay, so to summarize, scientific studies of consciousness can be roughly categorized into big two directions, according to the question we asked. And about the mechanism of consciousness, our findings suggest that, well, it seems that most of the neurocorrelates of visual consciousness can be found in early visual area, suggesting that this is a crucial part for visual consciousness. And as for the consciousness, our consciousness knockout paradigm suggests that, well, consciousness is not responsible for many functions, including saliency or saccades or syntactic processing, sound shape mapping, and facial attractiveness. So these functions appear to be intact even when you knock out consciousness. And our future directions, including trying to predict people's behavior by brain reading, and trying to change people's mood and behaviors by inserting unconscious information. And finally, we're trying to build a consciousness meter, or at least trying to examine the possibility of building a consciousness meter. And these are the people in the lab I want to thank, and also thank you very much.